Okay. Hi, everybody. Thank you for watching online our review session for today. I'm Annalise, and I am helping to organize our second annual Jacksonville Brain Bee, along with Gabby Fiorino and some of our TAs who you'll actually get introduced to when they start presenting. So I wanted to start today's session with a little bit of information about the Jacksonville Brain Bee so that you know what to expect over the next few months between now and the in-person competition in the spring. So what is Brain Bee? It's a three-tiered neuroscience competition that happens at the local, national, and international level for students to learn about the brain. And during this time, we want to show you how interesting and impactful neuroscience and neuroscience research can be. And we also want to give you a glimpse into what careers in neuroscience look like. And these are just some photos from last year's Brain Bee. On the left is from our Jacksonville Brain Bee, and then on the right is from the national championship. So this could be you. So how does it work? So at the local level, we'll have our second annual Jacksonville Brain Bee in spring 2025. That's probably going to be around March or April next year. And that date will be determined and sent to your teachers, so they'll know about it as we get closer to the date. And it will have the 1 day in-person competition event in the spring. And then in the months leading up to that, we'll have monthly virtual sessions. And this one is the first one. And that's where you will be able to come and learn more about the material that you've been studying in the textbooks or with your classmates. And then it'll give you a chance to kind of familiarize yourself even more and practice the material that you've been learning. And then the winner from this competition will go on to the national level, and we can help offer funding to help you with the travel and stay expenses. That's what we did last year for Carmen, who was our previous Brain Bee winner. So at the national level, there's the USA Brain Bee Championship that will also be taking place next spring or summer. And then that champion goes on to the International Brain Bee, where students from over 50 countries are participating. So it's a really awesome opportunity to learn about the brain and also do something that's like pretty cool and challenging and something that you can talk to your friends, and it's good for college apps too, probably, if that's part of your planning too. So these are some photos from last year. So you can see we had some brain dissections that student could look at and kind of test their knowledge on neuroanatomy. And then we had our volunteers who were helping to judge the different parts of the exam. And then we had our top students who were then doing that last round. That's more of a live round judging where we go until there's one winner left standing, quite literally. So that was a fun round to actually host. So you might be wondering, who am I going to be learning from as I prepare for the Brain Bee? So from our side, during these review sessions and at the actual in-person competition, you'll be learning from Mayo Clinic principal investigators who are people who run research labs here at Mayo Clinic. And so these scientists will often be doing the guest lectures, so they'll tell you about their science, but also hopefully they'll also share like how they got to that position, what their career journey was like. And then we also have Mayo Clinic graduate students. So these are MD, PhD, or PhD students. And we have University of North Florida undergrad students. And then Carmen Felix, last year's Brain Bee winner, will also be helping. And you'll probably meet a lot of these individuals in the in-person competition. But along the way, we want to give a special thank you to our volunteers who have been helping from Mayo Clinic and from UNF, because they've been doing a lot of the work to set up the Quizlets that you're going to receive and run the review session and create the Jeopardy games that will be helping you to review. So they've been putting a lot of time into that. So special thank you to them. And they're excited to be here and to be sharing their love of neuroscience with you. So hopefully you enjoy getting to interact with them too. So for the format, we have monthly virtual sessions, as I previously described, and the one-day in-person competition. So the format for the virtual sessions will be starting with a guest lecturer. For today, I will be your interim guest lecturer. And typically we want you to learn more about their research interests and their career journey. And I put stars here for like as a reminder that you can ask questions. Like if you have a question about, well, how did you become interested in that? And why did you pursue this and not that? Please feel free to ask. That's kind of why we want to be a part of this, to help share our journey. And then after the guest lecture, there will be a review session just of the material that you've been reviewing for that meeting. So make sure you ask any questions that you had while you were reviewing, anything that was unclear. And a special tip that I have been doing ever since I was in high school was, as I review my material, I leave like a space at the top of the page or on another page where I just list out questions that I have. And then when I go to my review sessions, I make sure I ask those questions because often you forget. So that's a tip that I would recommend using if that maybe works for you. And then after the review session, there will be a Jeopardy game, which will be fun. And the person with the most points at the end of it will get a gift card from us, probably for Amazon. So definitely get involved in that, and you might get an Amazon gift card. Treat yourself with that. Um, okay, so that's for the monthly virtual session. Then for the in-person competition, that's going to be one day here at Mayo Clinic, and there are three phases of it. There's a written exam style phase with multiple choice and short answer questions. And then there's a lab exam style, which will be like photos of brains, and that's going to test more of your neuroanatomy. And then based on the those two phases, then the top ranking students will then be in an elimination style round until one student is left. But the top three students will get prizes, and everyone will be able to say that they participated in this event and in Brain Bee overall. Because it is a lot of work that you're putting into it. So you should take credit for it. Okay, so this is the schedule. I'm sure that your teachers have communicated this to you because they have this information. If you don't have this information, please ask your teachers and then we'll make sure to get it to you. So once a month, every Tuesday on Tuesdays from 5 to 7 p.m. And then here are listed the different chapters that you want to make sure to review before that day. So that's it. If you have any questions, please feel free to reach out to your teachers and they can reach out to me or Gabby, or you can reach out to us directly. And before I leave, I do want to share a little bit of my journey in neuroscience. As I mentioned, I'm a PhD student here at Mayo Clinic. So I was born and raised in Massachusetts, the land of Dunkin' Donuts everywhere you go. And when I was in high school, I actually did a high school internship between my junior year and my senior year. And that's the first time I ever did anything related to neuroscience. So I was looking at patient MRIs of individuals with schizophrenia or who were likely to develop schizophrenia. And I was measuring the size of the corpus callosum. And in order to do that, I used this diffusion tensor imaging. It's like a special kind of imaging that's based on like how the water molecules are reacting to the MRI. So they're able to kind of track the orientation of water in the brain to figure out what your neuronal connections actually look like. It's pretty cool. And so we could actually make these 3D tractographies using the patient MRIs to visualize the parts of the brain. And that was also the summer where I actually held a human brain in my hands. And so with all of those experiences, that was the summer where I fell in love with science. And ever since then, my life has been studying neuroscience. Um, so yeah, it kind of happened towards the end of high school. And then I went to college and I studied neuroscience and I was taking classes mostly for my freshman and sophomore year. And then in my summer between my sophomore year and my junior year, I did my first internship where I was working in what we call a wet lab. And a wet lab is, so there's a wet lab and a dry lab. A dry lab is doing research, but you're on a computer most of the time. So that's more like AI, bioinformatics, data analysis, etc. But a dry lab is where you're doing more experiments, kind of like with your hands, working with cells, doing like Western blots, different techniques that are like they're actually wet. Um, so this was my first wet lab experience, and I was studying protein aggregation outside of the brain and peripheral tissues. But that's where I started to get interested in protein aggregation. And then a little bit after that, maybe like 6 months after that, I started doing research at my college. I got involved in a lab on campus, and they were doing single cell analysis of neurodevelopment. So that's where I started to do more neuroscience research again. Um, and that was really interesting. And there was a lot of data analysis there, which was pretty cool. And that's where I realized that I really liked spending time in a lab. So that's where I decided that I wanted to do my PhD. But I needed more research experience before I applied for PhD programs. So I got a job um, doing research. And this is where I was studying protein aggregation in Alzheimer's disease. And I was there for about 2.5 years, really just learning techniques and trying to build up my skills a little bit more as I applied to grad school. And then now I'm in grad school, I'm doing my PhD. I'm studying protein aggregation in Parkinson's disease. And I honestly don't have a ton of data. So I don't have a lot of cool figures to show you for that, but I do have a pretty photo of my cells. Yeah, so that's my journey through science. It took a while to get to, but each stage along the way, I learned a lot about what I like and what I don't like. And my focus kind of narrowed over time, just naturally. And I never knew that I was going to be so interested in neuroscience until really that first internship that I did when I was in high school. Okay, so last thing for me before we move on is my favorite parts of neuroscience. I think my favorite thing about neuroscience is that chemistry and physics, like when you're, and these are things that you'll learn in kind of like the basics of neuroscience, like the charges of ions and ions going back and forth across the membrane leads to the action potential of the neurons, and that dictates our emotions and our actions. So, a lot of what we think and how we feel and how we move is really chemistry and physics. And I think that's like such a cool aspect of science. Um, there's also a lot of potential to help others with your research in neurodegenerative diseases. So I think a lot of us who work in a lab and who you'll probably meet along the way will feel similarly about that. Like we are excited to make an impact with the work that we're doing. And as you saw from the last slide, it's a highly visual process. You know, you just the nature of the work, I think there's a lot of very visual elements, and that's something that I enjoy about my day-to-day work. Okay, so that's it. It was really nice to meet you all virtually. And with that, I will end the presentation and I will let our volunteers for the week, Ejlal, Emily, and Joy, who are Mayo Clinic students and UNF students, who will run the review session for today. And you guys can take it over and introduce yourselves, etc.
I just before we get started, I just also wanted to say great job, Annalise, for putting all this together and for everyone of all of our volunteers. And also for all of the students, I just want to emphasize Annalise's points of, really the great exposure you'll get to neuroscience. And through these meetings, we plan to have some guest lecturers, which will be actual scientists discussing their work and talking about their path to becoming scientists. So, please join these meetings because these are, scientists that are taking time out of their day to really join and teach you guys about what this life will be like. And we'll also, in addition to the competition, we'll have some career panels and some opportunities for you guys to really learn about the career and how to get involved in a career in STEM. So, just want to highlight everything that Annalise said. So yeah, thank you. Our TAs, will take it away.
I'll pull up my screen for the presentation for the review session. Are you able to see that?
Yes, we can.
Okay. So our topic for today is going to be brain basics and neurotransmission. And before we get started, we wanted to introduce our team, just a little bit about each one of us. So Emily, if you want to go first.
Sounds good. Hi, everyone. My name is Emily Huey. I'm a first year PhD student at Mayo studying neuroscience. And to provide a little bit of background of my research experience is in college, I went to UC Davis and I majored in neurobiology. And then throughout my time then, I also did some work in labs, studying Alzheimer's disease. And then I worked for 2 years as a research technician studying rare neurodevelopmental disorders. And now here I am at Mayo, um, trying to explore more of what I want to study for my thesis work. But I would like to say that my first interest in neuroscience actually started in high school. So this is a great time to really learn what you're interested in and what you'd like to do in the future. And ultimately, after my PhD, I hope to be a professor one day. So it's nice to meet you all.
And my name is Ijlal. I'm an MD PhD student here at Mayo Clinic. I did my undergraduate degree in molecular and cellular biology in a university in Maryland, and then I moved to Minnesota for medical school. I actually did a year and a half between my undergrad and like my med school for research. That's around the time that I was trying to decide whether I wanted to do just a PhD or whether I wanted to do an MD PhD. And it ended up being MD PhD. So I've done 3 years of medical school, and now I'm starting my first year of PhD studies in immunology. And I'm interested in autoimmune encephalitis. So that's the bit where the brain comes in. But I also, like Emily, first found my interest in both fields, whether it was science or medicine, in high school in a program similar to this one. So if you have any questions or you're interested in medical school applications or graduate school applications, I'd be more than happy to help out. It's good to meet all of you.
Hello, everybody. My name is Joy Bakhtor. I'm currently a freshman student at UNF, and I'm majoring in behavioral neuroscience with a minor in chemistry. I, as a freshman, don't have really that much of a background on research or anything like that, but I am indeed interested. I find all these opportunities that are given, and it's helping me gain more knowledge about the path of being in neuroscience. And I really hope that with that, I can be able to get into medical school and do more work about that. And if not, then other types of sciences that are related, I'm sure.
I'm muted. A quick overview of what we're going to be going through in the presentation. The first part, Emily's going to go over the introduction, the basics of the brain and the nervous system. And then Joy will talk to you about neurons. She'll teach you about action potentials and chemical messengers. And then I'll close up the presentation with a discussion about drugs and addiction. And these are all based from the chapters from your 2 textbooks. So Emily's going to take it away.
Sounds good. And before I dive into this, we'd like to preface that this is just a brief overview. It's not going to be completely reflective and detailed as what's in your textbook, but this is at least a review, just to see what are the highlights of each chapter. So now to go right into it in brain basics and the nervous system. Well, first, what is neuroscience? Well, neuroscience is the interdisciplinary study of the structure and function of the nervous system. With neuroscience, we can answer questions related to cognition, learning, behavior, language, consciousness, as well as diseases like neural disorders. And what I'd like to highlight below here is that there's many subfields that comprise neuroscience, such as behavioral neuroscience, cognitive neuroscience, computational neuroscience, and also neuroscience itself involves many different topics, including math, physical science, engineering, all the way to also philosophy or pharmacology. So it's really interdisciplinary. Now moving on to the next, let's talk about what is the nervous system. Well, the nervous system is the command center, and it sends messages that are electrical signals between the brain and the periphery. And it's used to regulate a variety of processes. So within the nervous system, we have our central nervous system, which is comprised of the brain and the spinal cord. And then we have our peripheral nervous system, which involve our sensory nerves as well as our motor nerves. And in the figure below, the nervous system is so important in enabling us to detect and react to changes inside and outside the body. So for instance, if we're about to touch a hot stove, our nervous system is what tells us, oh, let's not touch that hot stove. So now let's dive in deeper to what is a neuron. So the functional unit of the brain is the neuron, which are nerve cells. And these are fundamental units of the brain and the nervous system. What do neurons do? Well, they're responsible for sending, receiving, and transmitting electrochemical signals throughout the body. The classifications of different neurons are motor neurons, sensory neurons, and interneurons. And what's fascinating is that what we found in research is that there's approximately 86 billion neurons in the human brain. And to the right, we have the different parts of the neuron. So at the top, we have dendrites, which receive the signals from other neuron cells. We have our cell body, which is also known as the soma. We have the nucleus that contains the genetic material. We have our axon, which is what conducts these electrical impulses along the neuron cell. And we also have this thing called myelin, which insulates the axon, which also helps speed up the transmission of the electrical impulses. And lastly, we have the axon terminal, which transmits electrical and chemical signals to other neuron cells and effector cells. So what are the major landmarks of the brain? Well, the largest part of the brain is called the cerebrum. The cerebrum is made up of 2 hemispheres, and these hemispheres are connected via the corpus callosum. The cerebral cortex is what the deeply folded layer of nerve tissue is of the cerebrum. So what are the lobes of the brain? Well, there are 4 major lobes of the brain on each of the 2 hemispheres. First, we have our frontal lobe, which is here, which is involved in voluntary movement, speech, memory and emotion, the higher cognitive skills, problem thinking, and personality. We also have a parietal lobe, which is around this section here, which integrates the sensory signals from the skin, processes taste, as well as some types of visual information. Then we have our temporal lobe, which is down here, which is important for some visual processing, interpreting auditory information, and it also houses the hippocampus and amygdala, which are part of the limbic system. And lastly, we have our occipital lobe, which is in the back, which recognizes colors, shapes, and integrates them into complex visual understanding. So how does information travel within your brain? Well, information moves from one region of your brain to another via chains of neurons that can transmit signals over long distances. First, we have our nerve tracts, which are neurofibers of region-spanning neurons from distinct bundles, such as the corpus callosum or the anterior commissure. We also have neural networks, which are a group of these nerve tracts that connect a series of regions in the brain. And lastly, we have neural circuits, where each region of your brain analyzes a specific subset of all the information received. And these neural circuits enable that connectivity from each region to another. So to close off my section, let's talk about brain waves. Well, this network activity, as we talk about with the electrical signaling, creates brain waves. So as neuronal signals loop through the thalamus and the cortex, they produce these rhythmic, oscillating electrical patterns that can be detected with an electroencephalograph. So these signals that we observe here in this picture are commonly called brain waves. And there are 4 distinct types. What we can observe is that they are also very characteristic in this EEG layout. So we have alpha waves, which originally mainly are from the parietal and occipital lobes. And this is when your brain is relaxed, your eyes are closed, and these frequencies are from 8 to 13 hertz. And then we also have our beta waves, which are produced typically from the frontal and parietal region. And these are ranging from 14 to 30 hertz. Then we have theta waves, which are typical of sleep. And then delta waves are also characteristic of deep sleep. So these waves are really important because they show the brain activity as well. So it's really cool to go from the cell, the neuron, to a larger picture of whole brain activity. Now Joy is going to take it away and talk about action potentials.
Hey everybody, so neurons and the action potential. Something that all neurons have in common in their activity is that they're both electrical and chemical. The chemical signals that are received in the dendrites from the axons that contact them are then transformed into electrical signals, which then they add or subtract from electrical signals of the other synapses, which then makes the decision about whether it should be passed on, the signal, or elsewhere. The next slide, please. A neuron parts are in constant motion, and it has a concept called polarization. Now we're here looking at a diagram, but the ones that we're going to focus on is going to be the axons. We're going to focus on the dendrites, the cell body, and the synaptic terminal. And what we are talking here, the concept called polarization, is because the information they process are supposed to go only one direction. Doesn't mean they always go one direction, but they supposedly go one direction. And then next slide, please. We are going to focus on the receiving and deciding. On the receiving side of the cell, the dendrites have close contacts with incoming axons of other cells, each of which is by a minuscule gap, and these junctional spots are called synapses. Most of the cells in the cerebral cortex are located on the dendritic spines. Communication between those nerve cells are called synaptic transmission. And when the dendrite receives one of the chemical messengers that has been fired across the gap, separating it from the sending axon, miniature electrical currents are then set up inside the receiving dendritic spine. And when those currents come into the cell, it's going to be called excitation. And when the currents are going out of the cell, it's called inhibition. And moving on to the next slide, we are going to, as you can see here in this diagram, will show you it will show you when the membrane reaches action potential, goes back down, and then starts all over again. So what's happening over here in this little diagram as you can see. You can see how it will goes, once it does the current that's strong enough to reach the threshold, it goes and reaches the action potential, and then it goes back down, repolarizes, and it goes to a period called the refractory period. And what happens there is just a brief period of silence where the membrane then recovers its explosive capability, readying the axon membrane, and then going back to a resting state to repeat this all over again. And moving on to the next slide, we are going to be talking patch clamping, which is a modern electrical recording technique, and its function is that it enables the study of the movement of ions through individual ion channels in all sorts of neurons, and then it makes accurate measurements of those currents in brains that are just like our own. And as you can see here in the bottom, we got a little illustration of it. And now moving on to the next slide, we are going to be talking about what a transporter is. A transporter, its job is to suck up the transmitter in the synaptic cleft, and then it clears it clears the chemical messengers out of the way before the next action potential. And then there's the lock and key reference, which the key being the attachment of the transmitter, and lock being the receptor, which is also known as the ionotropic receptors, and these cause the opening of an ion channel. And going on to the next slide, we will be talking about storage and release. The neurotransmitters are stored in tiny spherical bags that are called synaptic vesicles. The arrival of the action potential leads to the opening of ion channels that are led in calcium, which as you will see is calcium 2+. And here's a little diagram of how it works. And moving on to the next slide, we have messengers that are capable of opening ion channels. The main excitatory neurotransmitter in the brain is glutamate. And we're going to move on and explain inhibitory synapses in a definition, which is the activation of receptors that leads to the opening of ion channels that allow the inflow of negatively charged ions. And there are two inhibitory transmitters that we're going to talk about, and that's GABA and glycine. And among many of the messengers that are acting on G protein coupled receptors are acetylcholine, dopamine, and noradrenaline. Moving on to the next slide, we're going to talk about metabotropic receptors. And they don't contain ion channels, and they don't lead to the inhibition of action potentials. And what they do is that they are found in complex particles linking to the outside of the cell to enzymes inside the cell, which then affects the metabolism of the cell itself. And the action of the metabotropic receptors is neuromodulation. And you got here a little contrast between ionotropic receptors and metabotropic receptors. And you can see here how the neurotransmitter in the metabotropic receptors is attached to a receptor, goes on, and you can see the ion channel as it goes in and out. While in the ionotropic receptors, you can see how the neurotransmitter is attached to the side, while the ions enter the cell and propagate the signal, and then you can see how when it's opened and it's closed. And now moving on to the next slide, we're going to be talking the science of addiction.
Yeah, thank you, Joy. So I'll be talking to you about the science of addiction. Before I get into the main portion of this discussion, I do want to give a bit of a disclaimer. So the topics that are discussed in this chapter are pretty hefty, and they have repercussions far outside the scope of just the brain bee. I only have about 5 slides to show you today, so it's by no means comprehensive of what the addiction encompasses in terms of society, in terms of what individuals experience, even in terms of what this means for medical professionals in dealing with people who are addicted, because it is a disease and unfortunately it is an epidemic in our country. So this is a very small portion of what the science of addiction is. And I really recommend some of the resources that are presented in your book and some of the descriptions of how it works in the chapters. I think those are really helpful, and they provide resources in case you're interested to learn more. To understand the science of addiction, we first have to understand dopamine and what this neurotransmitter means in our daily lives. It's really essential for humans to be able to do anything within their lives because I kind of equate dopamine with motivation. In order for you to get out of bed in the morning, to go find food, to build a shelter, to protect yourself, all of that requires dopamine. So we really can't survive without this really important chemical. And on a normal day, we exist within the theoretical range of about 40 to 50 nanograms of dopamine. So a pretty small amount comparatively, but look how much we're able to accomplish just with that. On a really good day, like if you're having the best day ever or you're having your favorite food, for example, for me, it's cupcakes, that gives you about 100 to 90 nanograms of dopamine, hypothetically. Now, for an individual who takes, let's say, a methamphetamine, that increases the amount of dopamine in the brain to a comparative 1,100 nanograms. So imagine somebody reaches that level of dopamine and then tries to survive daily with just 40 to 50. It sounds impossible, and unfortunately, it is an impossibility for a lot of people and a reality for many of them. And if you notice here, the first time an individual has that substance, they may get that 1,000, that 10 times the normal amount. But with every subsequent use of that substance, it decreases, meaning the effect decreases until with the same amount that they took the first time, they're only getting 100 nanograms. And so the only reasonable conclusion they draw is that they must increase the amount of that substance to reach that original 1,000, that original feeling of euphoria that people experience. Then the question arises, how is it that these drugs are able to affect our brain chemistry so significantly? And the way that they're able to do that is threefold. The first is something called disguise and copy, which means that they disguise themselves as the normal neurotransmitters that our brain produces and then trigger dopamine release. Remember, just keep in mind that the whole function of this drug, the whole goal of this drug is to release dopamine. So number one, we disguise ourselves as neurotransmitters in order to release dopamine into the synapse. That makes sense. The second way that they do this is they block reuptake of the dopamine. So either you can increase release or you can decrease reuptake of dopamine in order to make sure that a lot of dopamine remains in the synapse. So what they do is they block that reuptake receptor, put a padlock on it, and keep dopamine longer, keep that sensation of euphoria present for a longer period of time. And then finally, again, with that increasing dopamine in the synapse, they bind to those synaptic vesicles that Joy just talked to you about and increase the release of dopamine. So again, 3 ways: disguise themselves, block reuptake receptors, and increase the release of dopamine by binding these synaptic vesicles. Now that we know the mechanism of how these drugs work on a physiologic level, we can ask the question, what fuels the addiction? Why does a person keep taking the substance, even though it may have detrimental effects on their social life, on their professional life, on their family life? And the answer is twofold. The first is tolerance. So when these drugs enter the brain, increase the release of dopamine, increase the dopamine in the limbic system, which is kind of the reward center of the brain. What we've seen is that with each use, the effect of that chemical decreases, the effect of the drug decreases. Like we talked about in the beginning, how can I get to 1,000 when every time I take the drug, I'm slowly decreasing? So the only reasonable conclusion is I keep taking more of it. So that's number one. Number two is withdrawal. The sensation that these individuals feel, and when they describe the sensation of withdrawal, is really terrible, not only emotionally, because you're trying to work with 40 to 50 nanograms, which is your endogenous release of dopamine. When you were taking that substance, you were working with 1,000. In order to avoid the feeling down when they're off the substance, they take more of it. That's number one. That's the emotional side. But there's also a physical side. So for example, with opioids, the withdrawal effect is quite significant physically, because if you imagine what the worst flu you've ever had felt like and multiply it by at least 100, that's what these individuals feel. They truly suffer when they're going through withdrawal. I mean, they have rigors, they're constantly shaking, vomiting, nauseous, feverish, and it lasts for a very long period of time because it takes a long time for that substance to exit the system. And they've already developed dependence. The easy answer then for somebody who feels emotionally awful and also physically ill is to keep taking the drug. So those are the two things that fuel addiction. I do want to talk a bit more about tolerance because I think it's a very important concept to understand. And I really like how this figure shows the effect. If we imagine each iteration of taking the drug on the graph as the first line of the graph, and then the effect of the drug as the second line, you can see how even though you're taking the same, this individual is taking the same amount of the substance, the effect decreases over time. And then physiologically, we see that the amount of that neurotransmitter in the synapse is maybe the same, and it binds to those receptors. But with beginner use, you have a pretty strong signal being sent up the neuron. But over time, the number of receptors decreases. The sensitization of those receptors decreases, meaning they no longer respond to that substance anymore, that chemical, in the same way. And even the ones who are there, the signal that they send up is not as strong. So there's a really important physiologic change that happens in the brain chemistry of these individuals when they are using these substances. And to wrap up, I want to talk about the individual drugs that are mentioned in your chapter. And I highlighted these in gray just because I had highlighted the drugs that disguise and copy in gray. And I thought it was really interesting to give an example of these drugs that disguise themselves as neurotransmitters in order to perpetuate their effect on the brain. This graph to the side here is from chapter 4, and it just shows you the percentage of people who have ever used the drug, nicotine, alcohol, etc., and then the percentage who become dependent, meaning, for example, for nicotine, 32% out of 76%, versus something like prescription drugs where it's 9% of people out of 13%. So you can see that's a very significant difference there. That there's a lot of people who don't use prescription drugs, who are not dependent on them, and they're not at risk. But of the ones who do, there's a high percentage of dependence. I think that's an important distinction to make between these substances. So nicotine neurotransmitter that it affects is acetylcholine. The effect of it is a stimulant. And I do want to talk briefly about the treatment options that exist. So number one is replacement products. So things like patches, nicotine gum. When people are in the hospital and they're dependent on nicotine, and of course, smoking is not allowed on hospital grounds. So what we're able to provide individuals is with nicotine inhalers. They kind of like asthma medication, but it contains nicotine, and they're able to use that. It doesn't contain as much nicotine, nowhere near as much nicotine as in cigarettes, but it does help tide people over until they're able to completely remove that dependence. We do also have pharmaceutical treatments for nicotine dependence, and that's varenicline, which kind of stimulates this, which binds to the nicotinic receptors and also prevents nicotine from binding to those receptors. So it helps with the withdrawal symptoms. With alcohol, the treatment for it is I think really interesting because disulfiram is a pharmaceutical prescription drug that we're able to provide people who are dependent. And what it does is it reacts with ethanol in the stomach and it causes a nauseous sensation. So when that person imbibes alcohol, they feel nauseous and therefore develop an aversion to it. I think that's a very smart way, ingenious way of dealing with dependence and creating this sensation of aversion so that people don't even think about reusing the substance. There's also a camprosate, which also binds the receptors at a lower level. And then amphetamines, which are stimulants. And unfortunately, we don't have pharmaceutical treatments for this. Most of the therapy that's long lasting is cognitive behavioral therapy. And again, I want to say for both amphetamines and cocaine addiction, and also for opioid addiction, this is not a comprehensive list. The actual treatment for or rehabilitation to get out of this addiction can take years. And many people are not able to complete it because it's very, very difficult. So this is by no means a comprehensive list. And then finally for opioids, which are definitely a subject in the news because of how pressing this issue is in our country currently. But we do have prescription medications to help with opioid addiction. The first is, of course, naloxone, which is the immediate antidote for opioid overdose. And then methadone and buprenorphine, they have the same mechanistic action as opioid drugs. And therefore, for people who are trying to remove that dependence, they're able to take methadone and buprenorphine, which are not as addictive, to remove those withdrawal effects, feeling sick, feeling down, feeling depressed while they're going through the treatment. So hopefully this is a helpful slide, just an overview of all of the drugs and their different descriptions from the chapter. Okay, that was the last slide that I had, and that concludes our presentation. Thank you very much for your attention and for listening. And also thank you to my team. We had a great time creating the presentation. We're really excited to present it. So thank you.
Yeah, thank you, everyone. So I guess we'll end there for today. And if you have any questions, then please feel free to reach out. And for all the students who are watching this after, we are excited to see you in person the next time we meet. So yeah, thank you, everyone. Bye.